Okay, so a little bit about the talk. Sorry, I am a little bit nervous. This is the first time I've done a talk like this live. I've done a few iNaturalist videos and they're uploaded on YouTube, but this is the first time I'm live. So I'm really happy to be here. Thank you very much, Wayne and Richard, for asking me to present. We are going to talk about iNaturalist, a little bit about social media, and being a citizen scientist. So, who I am, I grew up in Tasmania. Um, I remember the first time that I remember uh, seeing a fungi was actually something that I later recognized as Amanita muscaria. I was walking to school when I was in primary school, and my younger brother and I, we used to pick them up and throw them at each other. Of course, my parents told me that was very silly and that they were poisonous and that we shouldn't touch fungi. So some years passed on, and living in Tasmania, it's a very lovely natural place. You start to explore. I like to bushwalk and hike, and you start to see plants and fungi. And there's a lot of information about plants, especially in Tasmania, but there's not so much on fungi. So I started to look for information. I came across some Facebook groups, particularly the Tasmanian fungi group, and then I started foraging for food. Um, it quickly snowballed, and I became obsessed. And a couple of pictures here that you can see me being silly. But these are pictures of fungi that I've taken to work, and basically any chance that I will talk about fungi, I will. And colleagues, friends, and family, they all need to know about fungi as far as I'm concerned. So we've got, I put the binomial name, and here's the Dutch name because I actually live in the Netherlands, and I've lived here for the past 6 years. And so right before I moved here was when I was really getting serious and starting to collect fungi. And I bought a microscope and I started to look at other features. And I I up and moved to Netherlands, and so it sort of started again with the fungi that I knew. Of course, there's some fungi that we know occur in Australia that are from Europe, but it was a whole new world, and I found it a little bit easier because there's all this literature associated with European fungi. There's a long history. There's many cultures that know a lot about fungi. In the Netherlands here, it's still a little bit funny. It's still a little bit of that. There's a lot of people that are scared of fungi. The Facebook groups and the mycological groups aren't actually as big as they are even in Australia. And so I came here and I really wanted to continue my obsession. And so I started doing excursions, showing people fungi. And basically, my aim with my excursions is to just get people really excited and to notice fungi. Um, I want them to appreciate them. And, out of like, if I have a group of 10 people, if it's just one person that's really excited and then messages me again later saying that was really great, I feel really happy about that. So, why iNaturalist? Well, it's a place to record, all your observations, whether it be fungi or any living organism. You can connect with people. I've connected with so many people here in the Netherlands, and it's been great for me because it's a way to find new friends. And I've connected with people, and we've gone out on forays. There's a person that connected with me recently, and they want me to help them with like excursions for proper foray with focusing on grassland fungi. So things like that. You learn from experts because there's people like Tom May, Matt Barrett, all these actual mycologists. Because I must say that I'm not a professional mycologist. I'm a amateur mycologist, and these experts, they have very particular expertise, and they can share something with you. which many have with me, that you might not easily find in a textbook, or you might not find in a research paper. So contributing to worldwide databases. So iNaturalist observations that reach research grade, they end up on the Australian ALA website, and they also will end up eventually on the Global Diversity website. I'm not quite sure. I think it's meant to happen both ways. And so if something's uploaded to ALA, I think it should be on iNaturalist, but I don't recognize that yet. But I certainly see records from iNaturalist on ALA. Improve your ID skills and species knowledge. We're going to work through some IDs later, and we're just going to explore. And basically, I'm going to show you how you can compare, how you can, search in the right areas for suggestions, maybe. And maybe you find a new species. Maybe you upload something and someone's like, "Wow, what's that? I've never seen that before." And maybe you're on the foray with the Mycological Society, and bam, there's something that's vouched, and you've found a new species, which is very likely in Australia. Let's face it. So some of the topics, we're going to look through the tabs. I'm going to actually just work on the website for a little bit, because I didn't want 50 slides of all the different things that we can look at. So we're going to do a bit of exploring. We're going to upload a mushroom, look at the identify tab. We look at the projects, and then we will look a bit further into some of the search functions, some of the tags and observation fields. I'm gonna continue with a few more slides now with something about taking the right photos, because uploading, a good photo, especially a cover photo, which I'll talk about more later, can increase your chance of getting the AI response to be accurate. And then we'll look at some Facebook things and some other cool things that we find. So, is there any questions? I can't actually see anywhere if it pops up. If there's questions, if there is questions, Wayne, maybe you can yell out.
Yes, I will do. I'll let you know if there's any questions.
So obviously we're looking at the back of someone, and we're looking at the top of a mushroom. I don't think I need to tell most people here. I think everyone's kind of experienced with going out on the forays and stuff, but we can't tell who it is. And it's the same case for the mushroom. Maybe if I'm related to this person and it's my brother, I could tell, and I could tell who this is. And maybe it's the same if I'm an expert in this genus of mushroom and I can tell what it is. But for all purposes, it's better to do something like this particularly. Let's go next one. Oh, so I've stolen this from Thomas, and I share it all the time because it's really useful. But it's a bit of a guide to documenting in the field. You've probably seen it if you're on the Facebook groups. Gives you a lot of information. I'm not going to read through it all, but basically, you can watch this back, because I think it's recorded, so you can watch it back. And save the picture, keep it handy, share it when people aren't providing the right information. And these aren't the best photos I found, but I just quickly grabbed some photos. And this mushroom here, tuberia, is actually the same as what I previously shown the top of. So now you can see just easily I have picked the mushrooms. Some people don't believe in picking the mushrooms, but if you want to accurately ID, it's really handy to pick a mushroom whole, with the base included. You want to take note of any features on the base, and it just helps. You lay the mushroom down next to something that's still in situ, and then all of a sudden you can see so many more features. Excuse me. And so this one's a tuberia, this one's an agaricus, if you can't tell. On iNaturalist, you can zoom in on this photo. You can see that the lamellae are probably not attached. You can see there's no color change. You can see colors, you can see remnants here. I hope everyone can see my cursor. You can see remnants here from the partial veil.
Yeah, we can see a cursor.
Cool. And so basically that just instantly increases your chances of what the AI will pick up when you upload your observation. This one's a funny one. I thought I'd add this in because just last week, I think it was, or the week before, this person posted this really strange mushroom to Facebook, and I sort of recognized it, and I was like, I think I know what that is, and I initially ID'd it as Girofragmium ininquins, something like that. And it was something that I'd only ever seen once before, and it didn't quite look like this one, but I'd done some reading and checked out some other observations, so I suggested this. And then I asked the person really nicely if they would add it to iNaturalist. And here we go, they did. And so the person uploaded it. They took this suggestion, which is fine. So they used their photo and they looked at the AI suggestion and it gave some in the wrong phylum, I think. But I was cruising through iNaturalist and I saw it, and so I gave that identification because I think it's the best identification at the moment that we can give it. There's a bit of discussion. Some other people added some things, and I tagged Tom May, but he hasn't actually replied yet because he was the one that sort of taught me about that fungus actually in a comment that he made on iNaturalist. So I thought that was cool, and I do that all the time. If there's something really cool or something that's useful, I ask people really nicely, and most people do. If people don't want to do it, I'm happy to upload things for people, and I'll give you the credit for the photo or whatever. And all I ask is that there's a location. It doesn't have to be exact location, but a rough location. And of course, you can obscure the location, which we'll talk about.
Quick
This
question.
one,
Zoe was asking, someone on a forum the other day was talking about the need for a photo 10 paces back to get environment. Is this important for iNaturalist? Do we need a habitat shot in the photos?
I always say that the more photos, the better, even if you upload 20 photos. But habitat, yes, definitely. If you don't know the tree species or the type of habitat, there'll probably be an expert there that will recognize something. And of course, if you find something that you suspect is mycorrhizal and you want to put the tree species up as well because you don't know it, you make a separate observation, and it's highly likely someone will give you the correct identification.
Okay. And I also heard that the first image is the one that the algorithm or the AI runs over. Is that
Yep,
correct? Image
yep,
one?
definitely. Yep.
So image one shouldn't be the habitat. Image one should be the specimen.
I tend to upload image one as my group photo. So if I get something, if I get a bunch of mushrooms and I can display different features, that's my cover photo.
Great, thanks.
Um, I'll show you later in the identify tab that you can actually toggle between the photos and then still compare using the AI. But if you're just uploading on your phone or if you're uploading on the uh, the computer, it just recognizes the first photo. So skip past that. This one, this slide's a bit about the observational fields in it, and I'll admit it's a little bit hard to find these observation fields sometimes, but I'll talk about that a little bit more. Something else, I'll skip past that. This is another one actually. This one will be fine. One of my topics was the rare and unusual finds and the cool things that I come across. And I saw this observation and I looked at it and I said, I know this mushroom, and I just couldn't at the time remember where I'd seen it. And I did a little bit of digging, and it was actually from Roy Halling's talk for Fungi Map. And I went through the slides. I'd taken a couple of slides from his thing, and bam, there it is. And I thought, wow, this is something that we can actually identify, and it's got a name. And so it hadn't at the time been uploaded to iNaturalist, so I requested the species be added, and then I identified it, and I tagged Roy Halling, who described it, helped. I think he described species, and he said, "Wow, yes, that's right." And you can see here some features that you can see. There are some more photos. I know I can link the observation later, but you can see that there's this color change here, the cap, and oh, not ready. You can see some blue staining here, and there's some comments. It's very typical of this. This is one of the holotype specimens. It has this marginate rim, and it's Velobelitis limbatus, and limbate or limbatus is referring to the color here between the two, the stipe and the base. Oh, I'm very good at accidentally tapping on this mouse here. And even though Roy collected it quite a few times, and he mentioned that it's widespread, it's only got what's that, 9 observations. So I'd consider this one not very common, even though Jeremy, who observed this, told me that he found it in a very popular walking spot. We haven't seen it again. And it's interesting because there's probably more species in this genus. There's other observations that Roy has uploaded that has similar characteristics, and DNA has been performed, and they are in the same genus most likely. And there's also specimens from Western Australia, so it could be one of these genus that's maybe more widespread than we realise. Um, During the week, Fran actually posted this little quiz onto one of the Facebook groups, and I'll admit I didn't really know what it was. I had some ideas. I thought, well, I think it's probably something associated with Marasmius or Marasmini. And I went searching on iNaturalist to help work out what it was. And so I was working through the suborder, and using some of the clues, I narrowed down that it was something that wasn't very common, and possibly only had a few observations, if there was any. And I came to 3 genera, and then I investigated a little bit more. And then Fran mentioned the spore shape being the giveaway, and I'd already sort of looked through these 3, and I'd noticed that one had very particular triangular type shaped spores. And so I did a little bit more looking. I didn't find out if they were enameloid or not, but I just took the punt, and that's how I came to my identification. I used iNaturalist. And so that's just one way that it can be handy, like it does take a little bit of expertise, because if you're just looking blindly, you maybe not be able to find what you're looking for, but we're going to work through ways on how you can actually find these things. So I found that it was actually a tetrapigros, and as Fran said here, she's really excited about it, and this, and I'm really happy that she posted this on the Facebook group, because I hope that other people see it and they get excited too. This one. So in Australia, obviously, we've got a lot of introduced fungi, and you can tell the introduced fungi by this little pink triangle. So if you see, when you're looking through the species, you'll see here in the corner, it'll actually say IN, or it'll have a little pink triangle, and that means that it's an introduced species. You can also edit species. So I've found here that Tricoloma terrarium wasn't its status wasn't known, so I've gone in and where is it here? I've clicked edit, and then this has brought up this one here, and establishment means I've clicked edit, introduced, and my tab bars come down, introduced, and of course if it's a native fungi, we can click native. If it's endemic, it's a little bit more difficult. There's probably a few fungi we definitely know are endemic, but we probably share a lot with maybe New Zealand and Southeast Asia as well, especially in Queensland. Um, okay, I'm going to switch at the moment to my browser. And right now, I'm not going to do my login and sign up and everything. If people don't know how to log in or sign up, I do have a video on YouTube that you can check out, but it is really easy. You can sign up with your Facebook or iTunes account, whatever it is. Um, very easy. So here at the moment, I'm just on the Explore tab. If I click here on this iNaturalist, it brings up my personal dashboard, gives me a bit of information. Same thing here. These are all my personal links. So you'll see here in community, there's people, projects. This will open up the all the projects tab. This will open up all the journals for everybody and the forum if you want to talk about other things. But these will give me my direct link. So if I click calendar, it'll show me a calendar of all the days where I found, or all the days where I've observed things, mostly fungi. This will say the IDs that I've given.
Quick
There's a little
question
bit.
for you, Shane.
Yeah.
Um, if I've got a specimen that I collected 5 years ago and I'm adding it to iNaturalist today, what's the issue there? Does it record it as a today recording, or can I edit the date?
Yes, you can edit the date. So you can edit if it's maybe you took it on an iPhone and that data's probably still stored. So we'll already automatically observe the date.
Right,
And when you upload it, I'll just click on this quickly. And so when you upload it, it'll still upload and show that you've uploaded it today, even though it says 5 years ago. So you can see here we've got 8 hours ago, 1 month ago, 18 days ago, 18 days ago. That's being shown in order of being uploaded. So these are all just been uploaded. If I hit this filter button, I can change by the date added, and I can change to date observed. And then all of a sudden, it's back to just order of when it was observed.
right, very good. I'm aware that some people might have a backlog of stuff that they want to
Oh
add,
yeah,
and I wasn't sure how that landed in iNaturalist. So thank you for
yeah,
that.
yeah. And I do encourage people to upload older observations. I see people uploading things from 20 years ago, so it's no issue. And it'll always come up in the recents unless you've filtered the date observed. So it is best to add to leave it as date added, and then it'll show everything that's uploaded, in the last today, basically. Okay, so we're looking at all organisms, and you can see there's 10 million observations in Australia. Let's look at Queensland. And this is still all observations, 2 million. And if we come here and we filter, we can see we've got some categories, birds, plants, all spiders, everything else. Of course, this little one's our fungi. And this picks up every fungal organism. So it's also slime molds and things that aren't traditionally, considered what we think is fungi. We've got some other things here. We can show, things that are wild or captive. This isn't too important at the moment. I can show you how to make something that's wild or captive, but it's it's not necessary in the terms of us searching at the moment. We can search things that are only research grade. We can search things that need ID, threatened. That's the same thing as It's not necessary at the moment because the data isn't really behind a lot of the species of fungi in Australia, and some are maybe wrong. For example, there was Morchella esculenta, which is a European species of Morchella. Um, it's been falsely recorded, most likely in Australia, early back even in the 1900s, because it's similar and it's a species that occurs down on the peninsula down in Victoria on the sand dune area. And so there was a couple of observations, and someone had made it that it was a threatened species in Australia because it wasn't a very common find. But it turns out it's not actually Morchella esculenta, and you know it's actually considered on the red list of fungi in Australia, even though it's not correct. So things like that, and it's just the data isn't behind the observations. We can just look at my observations. We can look at if it has photos. Obviously, if there's a fungal, you can put up an observation without any photos, but for fungi, it's not very handy, and it's kind of defeats the purpose of using iNaturalist. Sounds, if you want to upload something like a frog or a bird. Here we've got description tags. Um, say, for example, they're blue butterfly. You can use pink. You can, if someone's, uses, for example, you can have a location like your last foray. If everybody used the tag as the foray name, if you click on that, you can see all the observations from the tag. Here, of course, you've got date observed, so you can change, you can range it, you can say I want to look at just the month of January, but I want to look at it over a few years. Then I can look at that, and I can see things like what's the most common fungi found in January, um, things like that. You can, of course, yeah, pick just the month. Um, when I when I first started looking, I'm like, oh, how do I look back all the way to like the first record of that what was on iNaturalist? Or like, how do I look back 10 years ago? It's kind of hard if I'm just on the Explorer and I'm toggling through. And so here you can do date observed or date added, doesn't matter. Date observed, I go for this one normally. And then you change this one to ascending. So this one shows some of the oldest fungi recorded in Australia and see in Queensland, sorry, and you can see here 55 years ago, 52 years ago, things like that. There's an agrossoby 45 years ago. And so if I'm just bored and trolling through things, you can see here's some uh, observations from Roy Halling. And I think this one's actually, this would be one of his collections that he's made. And you can see down here, observation fields. He's got his specimen number, his name, and the other people that have collected, the habitat, the fungus substrate or host, and grouping, or how many specimen he has. The specimen was kept, yes, and it's in Perth. I've identified this one. We can see here there's the map, and we can zoom a little bit out so we can see exactly sort of where it is. And it should show up also. No, it's not working. Back, sorry. And so that's one way to look at it. Here, so I'll reset all the filters. Just go back to fungi, Queensland. And so we've got 85,000 observations, and there's only 1,700 species. So I guess that shows, how hard it is to identify fungi and how maybe much more work we still have to do in Australia. We can see the species here, and it's broken down and it shows you the cover photo of each species, and it tells you how many observations. And it's no surprise that this one's the most common species in Queensland, because it does stick around a while, and it's probably one of the most common ones posted on Facebook as well. And so if I'm new to fungi and I open up, or I go on a fungi walk and I see these fungi that are, on grass lawns and areas in parks, and I think, oh, what's that? This is a good way to maybe just recognize something easily. Here we've got the identifiers. And so these are all the people that are giving their time to identifying. Sophia does an amazing job. Look at that. I'm nowhere near that. I think I've only done something like 4,000 or 5,000. I can't remember. But this is just for Queensland. So Sophia's probably got a lot more. And observers. So there's 6,000 observers, and that's pretty good. We need more identifiers though, and especially people that have maybe expertise in particular areas. Okay. We move on to an observation. And so I've preloaded these ones. This is the tuberia we looked at before. And we click on the observation to be able to add some details. And so if I click on this one, it's going to load up some suggestions. And it's not very confident because this isn't a very good photo. So it's suggested Lacaria lacata, which isn't correct, of course. But in the list is tuberia. But if we look at this one and we see what it suggests, Boletails gets out of the way. It does suggest tuberia, and the top suggestion is tuberia fafacia, which is probably what this is. Without microscopy, I wouldn't know for sure. So I've got two observations. When you upload a photo, it comes out one by one. And so if you've got an observation that's more than one photos, you can simply pull and drag and click on that one. Or you can also go select, and you can go 1, 2, and then combine. But these two are separate, and this one's not really important, so I'm just going to close it. Here you can see the date and time that it's been uploaded. And normally, because on my phone I have the setting to grab my information from my GPS, it'll automatically pick the location, but it hasn't picked the location here because I've uploaded it through my computer. So I do need to randomly pick somewhere. I'll just quickly, yeah, hit update. That's not correct, but it doesn't matter. And so I've still here got this photo here at the well, the one that I don't really want as my cover photo. I want this one as my cover photo. And so I actually did that wrong. I should have separated them and dragged the other one onto this one. Sorry, this one onto the other observation, and that would have made this photo my cover. The species
Can
name,
you
no.
can you click on it and select make first image? Right mouse click and click make first image? No.
I think that's one failing of mine. I need to actually do it again. But online, once the observation has been made, you can actually edit the observation and then make it the cover photo. Normally, to be honest, when I'm uploading, I upload over my phone because I take most of my photos with my phone. And while the app for iPhones isn't great for most things like searching and identifying, it's pretty good for uploading, especially if you've got all your information stored on your phone with your location and your times. It makes it very easily. All you need to do is add your notes. If this is where you want to put if it's captive or cultivated, your location, normally I'm pretty public with my locations unless it's something that's rare or maybe it's something that I forage for and I don't want everybody to know about. So I would obscure it. If it's something, some people will make it completely private, but it's not useful at all. It doesn't. It'll just probably give you the state or even the country in some cases. So it might not be really useful as an observation.
Quick
Here,
question.
yes,
Grace is asking, who can edit things like threatened or introduced status? Is it just anyone?
it is anyone. Um, I would hope that curators are checking to make sure no one's abusing it or adding silly things. But it's also a bit of a niche thing that not many people are probably doing. The reason why I started to do it was because I noticed when you look at iNaturalist New Zealand, it's very much organised and all the species that are introduced are marked very well. And I thought, oh, I wonder why it isn't for Australia. And I thought, oh, I wonder how to do it. And then I found that you can do it yourself, and I just started adding them when I see them.
Very good. And you've already just touched on this, but Peter Boronsky was saying, "Hi Shane, could you please show people how to make the location private?" And similar to you, she said, "I have stopped uploading my finds of edible species because of foragers using iNaturalist to hunt down edibles,
Yeah,
particularly in national parks." Yeah,
yeah, yeah, and it does happen. Um, Unfortunately, there's people out there that do that. I always tell people I do forage. Here in the Netherlands, we're about the size of Tasmania, and we have the population almost of Australia, and so it's not fair to take everything. And I tell that on my excursions. We don't typically forage on my excursions unless it's something that's really abundant. And unfortunately, compared to Australia, the Netherlands doesn't have the nature that Australia does, and so there's just not that many places anymore, especially places like old growth and stuff like that. So yeah, it can be tough. I don't recommend people forage in national parks. There's plenty of other places to do that. Um, yeah, that's
the
another
fines are
topic.
huge.
The fines are
um,
huge. Like, it's just not worth it.
okay, just as a side note, I've got Sophie at Tazza saying you can reorder your photos using the edit function. Just an FYI, I think.
Yeah,
Kylie Agnew
yeah,
Francis is telling us, I believe private doesn't even show the country.
yeah. I
So
think
there's that.
yeah.
And Alita is asking, can projects use my discoveries without me intentionally selecting them, or do I have to purposely add them to projects for that to happen?
There's two types of projects. So on one type, yes, they can automatically be used. So for example, this one here that I won't click on it. It's a Dutch plant and mushroom edible project. Excuse me. And I didn't sign on to that one. It automatically adds my observations of plants and mushrooms that are considered edible.
Awesome.
And so someone's made that
Penelope,
project.
okay.
Sorry.
Penelope is advising the app is good for uploading pics, location, and obscuring location, and the
Yep,
ordering of the pics is easy to do.
it
Martin
is.
Martini saying, "Going well, big man. How obscure how obscured is obscured?"
Um, I believe it's a 5 or 10 kilometer radius, and it'll pick somewhere random each time you click on the observation. And so it won't tell you any one spot, and it won't really give you an idea of where exactly it is. In the description of the location, um, if we just click on one quickly, oh, you can hear Martin. He's worried about his foraging. It
So,
would
so
show
not very
you.
obscured then.
No, no, this isn't obscured. I'll need to find one that is obscured. Let's see. We'll probably this one's no, it's not. We might not find one, and that might be a waste of time. But basically, yeah, it'll, if you look at the observation, it'll show you a circle around a pinpoint, and that pinpoint's never accurate. I've tested it and it's not accurate. But here you'll show, here it tells you, I mean, a little bit more about where it is. So if you really wanted to, you could go to something like this area and start looking. Maybe you can find, maybe you could find the right habitat, but it's not going to be easy. So it's, I wouldn't worry too much about having your observations as obscured and then having people turn up at your locations, unless you put something else in your photos, maybe that's really identifiable.
Yep. And Alita Bond has given us an example of it in the chat with the
Yep.
circle around it. So we can see that. Yeah.
Cool. Thank you.
Thanks, Alita.
So we'll talk a little bit more about projects, I guess. I'm not sure, does the Mycological Society have a project? I searched, but I couldn't find one.
No, it doesn't. We generally use
Uh,
Fungi Map.
yeah. Okay, cool. Because I noticed that you do upload and you use the observational fields like I mentioned. Here you can your substrate, if you've got a name that you're sort of calling a recognizable fungus, maybe there's some DNA, ITS information that you can then attach to the observation, specimen kept, all that sort of information. And so it's, I guess for me, like iNaturalist is really about tying together all of these things and having a record where you can easily find this information. You can, some things that have been sequenced, DNA sequenced, they then have the gen bank accession. They have all this information about where the specimen is kept. So if someone wants to study it further, maybe there's a name that they have or a nickname, because it's not suggested to use something that is a scientific sounding name. So if you have an undescribed working name, you should use something that's like a nickname, like, or a color or something like that. And it's just something where you can tie all this stuff together, and then you've got photos to back it up. And I think, in this day and age, the photos and having that information, it's so handy. You read a book and you can see one photo of a species usually, and you can have the description, but then, there's all this more information that we can add to it now. If I've observed this same location, this same species, I can add a link to that other observations of the same species in the same area, for example. So it just helps when someone maybe is studying these things and they're searching and they want to come along and they go, oh look, there's a DNA sequence. I know that it's been vouchered. This is really useful. I think everything there. Sorry. No, if nobody, I've talked on this too much. So if anybody has anything else about that, just email me or chat me. It's fine. Projects, of course, there's the fungi site project, which is really cool. This is one of the ones where you do have to sign on, and I think Grace is listening. If I've gone too long too, please tell me.
No, you're right.
So we can click here, we can see the observations, the species, who's observing. We can add an observation. We can export some data, we can see some lists. So this one, you do need to sign up, and you do need to add some information to your observations. This one's cool, so I'll click on this one. Got this really big, thick partial veil and more hanging off the margin here. But we can go down here and we can see that it's in a couple of projects and fungi site, and we can see the observational fields. Here we can see, a bit more information, and these are all required. So if you don't fill this in, I don't think you can actually add it to the project. So it's something to consider if you see this project and I think, oh wow, I want to be part of this. Take note of these observational fields so you can go out in the field and actually remember to collect them. Now, if I click on this, I'll click this one. I can a tab will come up and it will says I can view observations with this field and value. I can with just this field, so just the key features, or I can do it without the field if I'm trying to exclude something. And so if I click on this one, brings up all the observations with that field, and there's only that one, but that's not a good example. Just quickly, PowerPoint. Okay, so this is a example here where I've clicked the observation field and I've come up with all these observations where the same field and value has been used. So I've used the undescribed working name, Cantharellus pink, because it's this seemingly fairly unique pink chanterelle, which is a little bit more robust than Cachinus, and it's mostly in the southern half of Australia. I think there's some from Adelaide or South Australia, Victoria and Tasmania, maybe up into New South Wales. But a bunch of us think that it's probably undescribed and it's pretty unique. So we've given it this way to be able to search, and we click this thing. The only thing is I haven't found a good way to actually search for the values. So I haven't found a good way to actually if I'm searching in my Explore tab, I haven't found a way to filter out. But the only thing that I have found is a bit of a link, and I'll send through a bunch of links because I have some links in my PowerPoint, but it's obviously you can't click on the PowerPoint. So I'll send an email through with a bunch of links if anybody's interested, and maybe you can distribute that. But this one's a bit of a menu on how to.
Quick question, sorry.
Yep.
Padmarupa was asking, what if you don't know the tree? And I think you touched on that earlier. That's the importance of having a habitat shot, isn't it? If you don't know the
Yeah.
tree,
Yeah. If you don't know the
just
tree,
include the habitat,
some
or
things
even
to add.
post the tree,
Yeah,
as
yeah,
a separate observation. Yeah,
yep, yep. Make a
right.
separate observation. If it's something that's important to you, make the separate observation. Obviously think about, okay, we're uploading fungi and we've got to look at some features to be able to identify that fungi. So think about the same with the tree. So maybe you need the bark, your leaves, something like that, if it's got a flower. So
Very good.
apply those same things to your other observations. I, I don't,
Grace Boxshall is also advising as
yeah,
well too, by the way. Another active research project on
yeah,
iNat is the Mullany barcoding project.
yep. Mullany. So is that for a certain area? It must be.
What proportion, Patrick Leonard is asking, what proportion of Queensland macrofungi can be accurately identified to species by participants in iNaturalist?
I'm totally just guessing, but it wouldn't be a large amount, I don't think. If we can go by, here with these numbers on Queensland, we've got 85,000 observations and only, 1,700 species. And if we look at the species, we go down pretty quickly we go from 1,450 and it goes down fairly quickly to 98. And so these are all only the species. Of course, if we're looking at observations, there's going to be all things that left at different like family level or genus that we're just not sure on. And so a way to search those is a way to exclude, say, the species is if we look here on the tab and we pick a rank. And so we've got everything from kingdom all the way down to species to form and hybrid. If we pick something like the family as our high and low, we're only going to pick up the specimen that hasn't been identified past the family, and so that can be handy. For example, I really like boletes. I search out boletes all the time. I spend a lot of time identifying them, but if I just want to focus on ones that we don't know anything about, or nobody's identified past the family level, then I can use that um, filter, and then all I've got is the family level, and so there's a Boletaceae there. Um,
Very good.
but but yeah, I think
Sophie at TAS was also advising other projects are polypore fungi of Australia,
yeah.
also Australian fungi of the tropics and subtropics, and also Queensland mycology, other projects. And there was just a confirmation as well too uh, from Grace that Maloney barcoding one is for Maloney fungi only. Yeah.
Yeah.
Fungi on Magnetic Island is another project. Penelope's advising. Picks up all fungi photographed on Maggie Island. So it sounds like there's a few extra projects out there as well too.
Oh yeah, definitely.
Sorry to interrupt you.
No, no, go for it. So I've just done the search. So before I didn't mention, but before I was actually on my own personal list of projects. That only was showing what I'm part of. And so here, if I click community and I click projects, it brings me up on the bigger, broader page for everything. Here you've got some tabs here. You can pick the user, and I've picked projects, and I've just searched fungi. And then, of course, you'll find lots of things here. Fungi map. And if I added in Australia or Queensland, for example, you can find other projects that way. There is a really cool project which is I know Grace mentioned about the uh, Mullany project, but there's an Oceania with DNA sequences. So if we pick this one, and this is a project where the user actually has to add the details because it's a bit more advanced, but it all seems to be working very well. And we've got some observations here, and if we click on them, we can see that the DNA barcode has been added, and we could copy that and then put it into Blast and then find the results and see what it matches. Or we can click here on the Myco map, which is an American project. So after this was collected, the DNA was sent away, and someone has come back with the result. It wasn't the species that was on, so they had to add it to iNaturalist. You can get the raw data, but then if I click on the observational field data, I can see all the observations with this barcode. Oh, this is the world, so I need to click Australia. So if there was another matching barcode, it would show up, but there isn't. This is a really handy tab to use, and I'll send this through in my links. Just explains a few more details about using the URLs to get, maybe to show multiple taxa at the same time. I do like this, but the only thing that I found would be really handy for my naturalist if the two taxa had different colors on the maps. That would make it a bit more handy. And just, yeah, a bit more detail about how to search out things in the observational fields. This is about the substrate and host. I'll quickly just take a look at the Facebook website, the Facebook pages that There we go. It's gone away. And so two main ones that I use is the some people don't know, but you can search out things. Some people don't seem to know that or be able to find the search tab. Um, you know, I encourage people to upload to Facebook, but the thing about Facebook is that it can be difficult to find those observations again. So if you're someone that's uploading lots of photos to Facebook, please do also check out iNaturalist, I guess. And then the Australian one, the Australian and New Zealand fungi. And anyone, I guess if you're obsessed, you're probably in this, and you can see all these cool things like we do. Um, is there any other questions quickly at all? Is there anything I've missed?
No other questions at this stage, no.
Tags on fields. I'll just quickly check my notes. Does oh sorry, I did open up another tab. There was a question right at the start about the edibility.
Oh, yes, and there was a link. Have you got the link?
I have. And so I assume it's this Ostroboletus. And I don't know anything about the edibility, I'm sorry, of Ostroboletus. I do know that there's one or two that that doesn't seem to be working. Can I confirm it was the Ostroboletus?
No confirmation just yet. There is a question. What makes an observer, an LP is asking, what makes an observation research grade?
So when a consensus is met, so if I put up an observation and I ID'd it to species, and then someone came along and agreed, if nobody else disagrees, that's a research grade. Um, that can be a tough one because there are plenty of observations out there that are research grade, which are probably not correct because sometimes people just tend to agree without taking proper diligence, or maybe it's just people that don't really understand or know. So if you've got someone that then disagrees, it then takes another two people to beat the other two people. So you need three IDs of correct species to be able to make that research grade again.
Right. And just back on that edibility one, Alita was advising the most recent someone that's identified as B. Hayedanus, Hayedanus, Alitas
Oh yes.
Hayedanus.
I do believe that one is edible. I know some people that have eaten it, and it is formally it is Boletus at the moment, but it's pretty likely that it's a Tylopolis. Tylopolis, some are edible, some are inedible. They're not exactly toxic, but you probably won't have a nice time if you do eat um, the bitter tasting one. So if you took a spit test of the mushroom, so you took a little piece of the flesh, you chew it in your mouth, and then you spit it out, and then you observe the taste. If it's something that's bitter, with boletes in particular, not for all mushrooms. If it's something that's bitter, it's generally considered not edible. I do believe that one is a mild tasting one. So if it's something that's mild tasting, um, now I'm not saying apply this across the board in Australia because there are some certain unknowns, but here in Europe, it's very well considered that I can go and taste test any bolete in the forest, and based on if it's a mild, you know, pleasant tasting mushroom, then it's considered edible. Um, in the cases where they're the toxic boletes, you'll find that they have some taste or some smells that are recognizable that steer you away from the edibility of them. I've just clicked on this research grade one, and you can see here two people. Sorry, was there a question?
Mark Marshall was just asking, by the way, will you be taking tours next time you're in Queensland?
Yes, I hope so. I was there in January last time, and I did a few little excursions, and I did a couple in Tasmania. But I'm hopefully planning to come back in the summer here, so probably from June maybe till August. And if I can get away with it, yes, I will. I won't be doing paid excursions or anything. It'll just be something that I'll probably announce a day or two before, and then see who wants to show up and get excited about fungi. Because for me, it's also me exploring and finding new things. My obsession really grew when I left Australia, and now I'm sort of living vicariously through everybody on iNaturalist and Facebook, and I guess that's why I'm so into it, because I do miss Australia, and I miss the fungi that are there. There's some really cool and unique things, and there's also just so much still to find out. So I will, and I'll post that probably to my Facebook page, the Tasmanian Forager. Um, and yeah, people can just message me and sign up when I do.
Awesome. Grace Boxall is just adding, by the way, as well, too, that as you're talking about research grade, she said, I understood that usually an observation requires two identifications that agree with each other before it goes to research grade. If there's disagreement, then it takes more. So she's agreeing with that.
Yeah, perfect. So I can agree here, and then that makes that a pretty solid identification. But if I didn't agree and I wanted to check some of the data, you know, I can downvote some of this stuff. So if I didn't, if it was a mushroom that was maybe typically found in summer, but it was the middle of winter, maybe you could question whether the date is accurate, or something like it shows a picture of the forest, but the location is at the beach. So you could, is the location accurate? Yes or no? Maybe it's something that someone's cultivated, and they haven't considered this, so you can put, and it's obvious that it's cultivated. You can downvote that it's wild. It's not wild. People. I've seen the funniest things, like, for example, someone uploaded a really bad photo. Apparently, there was something in the photo that was a living organism, but to me, all I could see was a piece of carpet. And I thought, well, what is this person doing? And someone had identified it as a lizard or a fungi. I think it was a fungi, and that's why I'd come across it. And I thought, well, what is this? And I thought, well, there's no evidence of organism, so I downgraded this. And then, instead of being able to be something that's research grade, it puts it to what's called a casual observation. And so it's something it's not considered useful for scientific research, basically. But you know, so
Awesome.
it's not. What's that? Sorry.
Awesome.
Cool.
That's great.
Um, I did want to show one more thing, just on my favorite. So if I see something really cool that I I normally like it, or I favorite it. And you can see at the moment there's a lot of boletes in there, and this just sort of saves me. Maybe it's something that I'm interested in and I want to research more, or it's something that I think is, for example, here, I think it's identifiable macroscopically. We don't have any idea what these are, these two boletes. And I've actually observed this myself in Queensland when I was there last, and Jeremy has as well. And we're fairly certain that if this bolete had a name, maybe it has an old name that someone else described years ago, and we just can't find the correct description. But we believe that this is very identifiable, and we could probably give it a nickname to make it searchable. But this is some of my favorites. Now we've got something here like Roy Halling actually just uploaded yesterday or the day before a whole heap of what he's calling xanthoconium, and I've been collecting these ones here because I thought the same thing. I thought, some information that I'd read about some Asian species. I thought, well, these look very, very similar, and so I'd started favoriting these here, and so I can start collecting a bunch of them. And then if someone comes along and gives them a name, maybe there's a research paper, I can go, oh, here we go, I can name all these, and it's
Grace
not
was asking
digging
how
through.
do you, Grace was just asking how do you designate a nickname?
Well, I've actually only designated one or two, and it's just been, I mean, the Cantharellus one was a fairly easy one. I thought it was pink. It's a recognizable color. Um, it does suggest when you are thinking of a name not to use something scientific sounding. Um, maybe that's in one way to protect you if you're publishing the name. Maybe someone else will pick up the name and use it. But also, yeah, I think that's probably maybe the main reason. Um, I think, did I cover
Great,
that
thanks,
question
Shane.
properly?
This has been, yeah, I think you did. It's been really great.
So yeah, I think in Australia, obviously, there's a little way to come. Boletes and agaricus are some of my favourites, and we've got people studying both groups. So I guess I'm pretty excited when a new paper drops, and if there's good examples and details about species, you can then go to iNaturalist and start trying to look for the species. I can if there's a location, for example, in the in the research paper that tells you where the mushroom was found, you can concentrate your search around that area. So I can look at map. And if I wanted to just create a box, say around the Cooloola or the Queensland sort of coast area, and then I can easily see all my fungi I found there. Maybe I'm particularly looking for a bolete. And then here's an example of the xanthoconium that Roy uploaded. And I think that this one's probably pretty widespread in Australia. They tend to have brown spores compared to the olive green sort of spores that Boletus SS has, like your Boletus adulis. And so if you recognize that this is still kind of brown, if this was something like Boletus adulis, it's already sort of turns more olivey green, a yellow to, pale to yellow to olive green. And Roy adds all this information about the species, so you can study this and you can think when I'm looking at other observations, use this information to confirm your suspicion of a species. Of course, he hasn't got a species here because nothing's been published yet, but it's really good that he uploads this stuff for us as references because this one's probably from 2009. And so I know that most of this stuff is on his website, but I'm not sure about other people, but I find the website really hard to navigate and the photos take a long time to load. But iNaturalist just seems to be really easy for when you're searching things and photo wise. Um, I think that's about it. If there's any other questions, if there's anything at all, shout out now or
Uh,
give me an email.
said this has been really fabulous, and Tammy said thank you. She had to leave, but thank you. It's been a really good talk.
No worries. Yeah,
Awesome. We
hopefully it didn't
appreciate
seem
you.
like I was too nervous or anything or
No, not at all.
fumbled my words too much.
No.
There's a bit of
Cool.
there's a few links here. Of course, most people probably know about the Facebook pages and stuff like that, but I'll send that all through in an email just so that it's easy to click on.
Send it out.
And
Recording.
thanks. I'm a bit of a silly person at times. My wife said, why you put that in there?" But it's like a drug almost. I get really excited. Mark that asked the question before, he's actually my brother. And I really distinctly remember once we were on a bushwalk and he said to me, he said, "Look at that tree. It's massive. Let's go. We've got to go and touch the tree." And so we walked up to the tree and I walked around the tree and lo and behold, I found my first ever Fistulina spiculifera. And I'll never forget that moment. And then like this one, for example, this Hericium coralloides, it's quite a rare species here in the Netherlands, and I just found this massive fruiting of it on this beech tree. So yeah, I enjoy a joke and whatever. Thanks for having
So I've
me.
got a thank you. I've got a bundle of
Oh,
thank
sorry.
yous coming through. Awesome presentation. Thank you. Thanks. Thanks. It's been really fabulous.
Thanks.
Thank you, Jen, for your generous use of your time. Thank you. Great presentation. Really good presentation. Thank you, Shane. Brilliant. Definitely learned some things. Appreciated. Thank you,
Yeah.
Shane. It's always a thrill to have you comment on one of my Baltic finds. That's brilliant. Terrific. Well, thank you so much for that. And
Thank you, and go and look for fungi and upload and get excited.